Namaste and welcome to Dr. Varsha Gaitonde's channel. In this lecture, we shall discuss some of the multiple choice questions related to genetics and plant breeding. If you are interested in answering the question, you just listen, or if you are the beginner, you can watch the slide and note down the answer. So let us move to the first question. The phenomena of like begets like is due to. The four options given here are genetics, heredity, germplasm and variation. The answer to the phenomena of like begets like is nothing but heredity. Transmission of characters from one generation to the next or from parents to the offspring is called heredity. Variation, recombination, mutation are the four answers given here, and the answer for transmission of characters from one generation to the next is nothing but inheritance, or it is also called as heredity. Variation is, options are differences between parents and offspring, difference between individuals of same species, difference among the offsprings of the same parents, and all the above. The answer is variation, means it may be any of the above answers. Means all the above is the answer. Term genetics was coined by Bateson. As you know, in the year 1902, Bateson coined the term genetics. Greek word which means to grow into. To grow into, the four options given are genetics, genesis, inheritance and factor. The term to grow into means genesis. Sixth question is, first scientific explanation regarding inheritance was given by Bateson, Mendel, Griffith, Johansen are the four options. The first scientific explanation regarding inheritance was given by Mendel with his peace plant experiment. Next question is, organisms produced by asexual reproduction are called clones, offsprings, factors, both A and B. Here offsprings are by the sexual fusion and factors are nothing but genes. So the answer is A, clones. Organisms produced by sexual reproduction are called as offsprings, clones, characters and genes. Answer is obviously offsprings. Offsprings are, the four options are exactly identical to either of their parents, not exactly identical to either of their parents, show intermediate characters inherited from both the parents, and both B and C. The answer here is both B and C, because offspring are not exactly identical to either of their parents and they show intermediate characters. They may show intermediate characters inherited from both the parents. Next is, term factor for gene was coined by obviously Mendel. Gregor Mendel was born in, the options are UK, Austria, Russia, Czechoslovakia. The answer is Austria. Mendel was a, options, physiologist, mathematician, cytologist, taxonomist. Mendel was a mathematician. First scientific study leading to the formulation of laws of inheritance was carried out by Mendel only. Under which title was Mendel's work published in Natural History Society of Brunn? The four options are Mendel's laws of inheritance, experiments in plant hybridization, experiments on heredity and variation, then origin of species. Origin of species is related to Darwin. Then this law of inheritance and heredity and variation, all these are Mendel's working principles, but the work was published with the name Experiments in Plant Hybridization. Mendel's laws were first published in the year, the options given are 1875, 1890, 1928 and 1866. It was in the year 1866. The year 1900 AD is highly significant for geneticists due to chromosome theory of heredity, discovery of genes, rediscovery of Mendelism and principle of linkage. See, here we can relate the question number 17 and 18. The rediscovery of Mendelism was taken place in 1900 by three eminent scientists, Hugo de Rice, Sermac and Karl Correns, who did independent experiments and proved the Mendelism. Mendel's work was rediscovered by three biologists from which of the following countries? The answer directly, I will move to the answers because if you will read all the answers, you will get confused. Austria, Holland and Germany. Mendel selected pea plant because it had a short lifespan, produced many seeds and large flowers, then many contrasting characters. In the above question, I just give you an exercise. Just mention which scientist among the three, Hugo de Rice, Karl Correns and Sermac, to which country these three scientists belong to. Okay, and the above countries like Austria, Holland and Germany are related to them only. Let us move to the next question. Mendel selected pea plant because it has short lifespan, then produced many seeds and large flowers, then many contrasting characters. All the answers are true. Botanical name of garden pea is Pisum sativum. Dominant character in pea is wrinkled seeds, inflated pod, terminal flower, dwarf plant. See, for wrinkled seeds, dominant character is tall, terminal flower, axial flower is the dominant character. For dwarf plant, tall plant is a dominant character. That means wrinkled seeds, terminal flowers and dwarf plant are the three recessive characters, and here the only dominant character is inflated pod. Character which was not considered by Mendel. The four options are seed coat color, wrinkled or round leaves, tallness or dwarfness, then position of flower. Answer is wrinkled or round leaves. This character was not at all considered, means leaf character was not considered by Mendel. Inherited character and its detectable variant is called as, the options are allele, trait, gene, both A and B. It is trait. Best describes a gene. Options given are triplet of nucleotide bases, specific length of DNA responsible for the inheritance and expression of a character, specific length of a single stranded RNA, both B and C. The best explanation to describe a character of gene is specific length of DNA responsible for the inheritance and expression of the character. Next question. Mendel's factors are in fact genes. When Mendel was defining, he was not knowing the term gene. Later on, Johansen gave this particular term to factors. The gene was coined by Johansen. Allele or allelomorph occur at same position on homologous chromosome, same position on heterologous chromosome, different position on homologous chromosome, different position on heterologous chromosome. The answer is same position on homologous chromosomes. That means alternative form of gene are nothing but alleles, and they are located on a particular location known as locus. Who proposed the term allelomorph? This was proposed by Bateson. Dominant allele means, the options given are allele whose effect is masked by another allele, allele that prevents the expression of other allele, allele without any effect, an allele which cannot express in presence of other. The answer is an allele that prevents the expression of the other allele. The external appearance of an individual for any trait is called as phenotype. Okay. Genotype means genetic constitution of an organism. Homozygote individual, options are breed true to the trait, does not breed true to the trait, then produce only one type of gamete. The answer here is both A and C, because homozygous individual breed true to the trait and they produce only one type of gamete. Term indicates a pair of dissimilar alleles. Options: homozygous, heterozygous, homologous, all of these. Answer is heterozygous. Cross between two pure individuals differing in at least one set of character is, options are monohybrid, polyploid, mutant and variant. Listen the question carefully. Cross between two pure individual differing in at least one set of character is nothing but the monohybrid, because here the pair or the cross is getting between the two pure individuals. F1 generation means the first filial generation. Question's answer C is the correct answer. Filial means the options given are offspring produced in sexual reproduction, offspring produced in asexual reproduction, offspring produced in vegetative reproduction, both B and C. Filial means the offsprings produced by sexual reproduction. As you might have studied in different breeding methods, like they'll cross A into B, they'll get the F1, they'll self the F1, they'll get F2. Likewise, when crossing is happening continuously, then the filial generations will go to different generations. Okay. Likewise, when it comes to offspring produced in asexual reproduction, they may be called as clones. Then offspring produced in vegetative propagation, they can be called as grafts or propagules. I think this is clear. Next question is F2 generation is produced by, answers are crossing F1 progeny with one of the parents, selfing the heterozygous progeny, selfing the parents, and a cross between recessive parents. The answer is selfing the heterozygous progeny. Means if you cross the F1 into F1, you will get the F2 generation. In genetics, the use of checkerboard was given by Mendel, Correns, Punnett and Darwin. Checkerboard is also known as Punnett square. Okay, so this was given by Punnett. Mendel in his experiments, options, maintained qualitative records, maintained quantitative records, conducted ample crosses and reciprocal crosses, and all the above. Mendel in his experiment followed all these things. He maintained the quality, he maintained the quantity, as well as made ample crosses for many generations, as well as reciprocal crosses. Before Mendel, or the predecessors of Mendel, were not successful because of the many reasons. Because Mendel was expert in mathematics, he was applying the principles of heredity and variation, and his vision was very clear. He was maintaining records as well as systematic generation studies. Along with that, he studied one generation at a time. Means he was not in mixing condition. He was not studying many characters at a time. Single character study made him the clear-cut explanations. To eliminate character chance factor, Mendel performed monohybrid cross, dihybrid cross, reciprocal cross and trihybrid cross. The answer is reciprocal cross. Mendel always started his experiment, that is monohybrid and dihybrid crosses, with any pea plant, heterozygous plant, pure line plant, fresh new plant. The answer is fresh new plant. Mendel carried out artificial cross by, the options are emasculation of selected female parent plant, emasculation of selected male parent plant, dusting of the pollen grain from selected male plant over selected female plant. Here the answer is emasculation of selected female parent plant. Why we do the emasculation? Emasculation is the process of removal of the male gametes, okay, or else we can call it as anthers we will remove from the female or a hermaphrodite flower. Then dusting the pollen grains from selected male plant over the selected female plant, because the desirable male plants are selected, from their pollen is collected and pasted on the female plant. Emasculation. What is emasculation? The answers are removal of stamens before anthesis, removal of stigma before anthesis, removal of petals before anthesis, and removal of sepals before anthesis. The answer is removal of stamens. Stamens are nothing but the male reproductive part, that is nothing but anther and filament together known as stamen. In pea flower, how many stamens are free and how many are fused? In pea, as it is a dicot, just like your red gram or pigeon pea or chickpea, it has 1 plus 9, 9 fused and 1 free. That is A answer. F3 generation was obtained by selfing the F2 hybrids. What result did Mendel obtain after monohybrid cross between tall and dwarf pea plant? The answers are all new plants were dwarf, all new plants were tall, 50% of the plants were dwarf and 50 were tall, 75% was tall and 25% were dwarf. In F1 generation, Mendel obtained all new plants were tall, okay, and in F2 generation, he got the segregation of 1:2:1, or if we talk about the phenotypic character, he got 75% tall plants and 25% dwarf plants. Next question. When Mendel allowed natural selfing of F1 hybrids during monohybrid cross between pure tall and pure dwarf pea plant, he found all plants were tall, all plants were dwarf, dwarfness reappeared in some plants, tallness reappeared in some plants. The answer is dwarfness reappeared in some plants. During monohybrid cross experiments, Mendel performed a reciprocal cross by selecting, the options are tall plant as male and dwarf plant as female, tall plant as female and dwarf plant as male, then both male and female plant as tall, fourth option, both male and female plants as dwarf. The answer is tall plant as male and dwarf plant as female. After performing reciprocal crosses between tall and dwarf plants, the ratio of tall and dwarf plants obtained was 3:1. Here I am giving some direct answers because there is nothing to discuss in multiple choice kind. Okay, Mendel grouped all contrasting characters in dash pairs in 7 pairs. Okay, the conclusion drawn by Mendel based on monohybrid cross was each factor exists in 2 contrasting or alternative factors. One of the form is dominant and another is recessive. Inheritance of each character is controlled by a pair of factors. Next question. From the reappearance of recessive traits in F2 generation, Mendel concluded that factors do not mix with each other in F1 generation. Factors remain together in F1 generation. Factor mix with each other in F1 generation. The answer here, the elimination is answer C. We can eliminate this because there won't be any mixing that is stated in the law of segregation and the law of independent assortment. Means there won't be any mixing. During gamete formation, law of independent assortment, here you need to remember one thing. It talks about a pair of genes or factors, but law of dominance is just about a single gene with 2 alleles. During gamete formation, diploid gametes are formed. Each gamete receives only one factor. Factor do not segregate. All offsprings show recessive characters. During gamete formation, each gamete receives only one factor or only one gene. Crossing of the homozygous tall pea plant and homozygous dwarf pea plant would yield plants in the ratio of all heterozygous tall. The crossing of homozygous tall plant and homozygous dwarf pea plant would yield plants in the ratio of, the answer would be 1 homozygous tall, 1 homozygous dwarf and 2 heterozygous tall. That is 1:2:1. Next question. Mendel crossed a pure white flowered pea plant with pure red flowered plant. The first generation of hybrid from a cross should show all the red flowered plants. That means first filial generation or F1 should show all red, because here red is dominant over white character. In monohybrid cross between pure tall and pure dwarf pea plant, how many type of genotypes are found in F2 generation? In F2 generation, we may find 3 phenotypes. Out of the 4 progenies obtained in F2 generation by crossing pure tall and pure dwarf, how many of them will receive only recessive trait from both the parents? Only one will receive recessive trait from both the parents. The monohybrid ratio is defined as, the options are: phenotypic ratio obtained in F2 generation of monohybrid cross, phenotypic ratio obtained in F1 generation of monohybrid cross, genotypic ratio obtained in F2 generation of monohybrid cross, and genotypic ratio obtained in F1 generation of monohybrid cross. The answer is A, that is phenotypic ratio obtained in F2 generation of the monohybrid cross. Next question, which of the following is phenotypic ratio of Mendel's monohybrid cross? Phenotypic ratio means, by closing your eyes, you should write as 3:1. If it is genotypic means 1:2:1. The law of dominance is illustrated in the garden P. Answers are heterozygous tall into heterozygous tall, heterozygous tall into homozygous tall. Then some others are given. Dominance means it is homozygous tall into homozygous dwarf. Okay. Mendel's law has not been proved to be true in all cases. The answer given is law of dominance. When Mendel proposed law of dominance, law of segregation and law of independent assortment, the two laws, that is segregation and independent assortment, were accepted by majority of the scientists, even though law of independent assortment has many of the exceptions, but law of segregation is largely and widely accepted across the globe. But law of dominance is a neglecting factor. Law of purity of gametes is nothing but law of segregation only. The second law of inheritance proposed by Mendel deals with independent assortment. Next question, reappearance of recessive traits in F2 generation is due to, the options are law of independent assortment, law of dominance, law of codominance, and law of purity of gametes. The answer is law of purity of gametes, that is reappearance of recessive trait in F2 generation. Some cases what will happen, in grandfather one character will be there, in father it won't be expressed, but again in the grandson it will be expressed. That means reappearance of some of the traits, that is known as atavism. This is for your kind information. Okay. Next question, which of the Mendel's law will always prove to be universally true in all cases? As I told you, that is the first law. The first law is dominance, second is law of purity of gametes, that is law of segregation, and third law is law of independent assortment. So here the answer is the second law, that is the law of segregation. Mendel formulated the law of dominance and law of purity of gametes on the basis of test cross, back cross, monohybrid cross, and dihybrid cross. The answer is monohybrid cross. A cross between two pure individuals differing in two sets of characters is known as dihybrid cross. Next question, the phenotype of plant with genotype capital Y small y, capital R small r must be yellow and round, because here yellow and round are the dominant characters over white and wrinkled. Next question, dihybrid ratio is defined as the phenotypic ratio obtained in F2 generation of dihybrid cross, phenotypic ratio obtained in F1 generation of dihybrid cross, genotypic ratio of F2 generation of dihybrid cross, and genotypic ratio obtained in the F1 generation of dihybrid cross. The dihybrid cross means that is the phenotypic ratio obtained in F2 generation of the dihybrid cross. That is nothing but your 9:3:3:1 ratio. Next question is, while performing dihybrid cross, Mendel, the options given are, selected a variety of pea plants having yellow and round seed as female parent and another variety having green and wrinkled seeds as male parent, obtained pure lines by selfing, performed artificial cross by emasculation. The answer is all the above are true. Means he followed a systematic procedure. Out of the 4 phenotypes obtained in F2 generation of dihybrid cross between yellow round and green wrinkled seeds of pea plant, the options are 2 were parental and 2 were new combinations, all were parental combinations, all were with recessive trait, all were new combinations. Here the answer is 2 were parental and 2 were new combinations. The statement probability of 2 independent events occurring simultaneously is the product of their individual probabilities is, the options are law of dominance, principle of probability, law of segregation and law of new combinations. Here the answer is principle of probability. What principle of probability states? It is probability of 2 independent events occurring simultaneously is the product of their individual probabilities. The next question is 3:1 into 3:1 is equal to 9:3:3:1. This signifies trihybrid ratio, 2 monohybrid ratio. Dihybrid ratio is a product of 2 monohybrid ratio. None of the above. The answer is this is a dihybrid phenotypic ratio with 2 monohybrid ratios. That means here 3:1 and 3:1 are the 2 monohybrid ratios. Mendel's pattern of inheritance systematically showed the progeny in checkerboard, squareboard, crossboard, all of these. It is Punnett square or checkerboard. The conclusions made by Mendel based on dihybrid cross was, when a dihybrid or polyhybrid forms gametes, each gamete receives only one allele from each pair. The assortment of alleles of different traits is totally independent of their parental combinations. Both these statements are true with respect to Mendel's dihybrid cross. When Mendel crossed pea plant with yellow round seed and green wrinkled seed, the seeds obtained in F1 hybrid were yellow round. The phenotypic ratio in F2 progeny in a dihybrid cross is 9:3:3:1. The genotypic ratio क्या होगा 1:2:2:4:1:2:1 and 2:1. Okay. New character combination appear in F2 generation of a dihybrid cross mainly because of independent assortment. Here how you can make this particular ratio, to make this you need to follow a simple formula. Just multiply this with this. That means in monohybrid ratio you were obtaining this as genotypic ratio. By multiplying these two you will get a direct dihybrid genotypic ratio. Okay. The next question is, new character combination appear in F2 generation of a hybrid cross mainly because of the independent assortment. Law of independent assortment can be explained by dihybrid cross and dihybrid ratio. Dash occurs due to crossing over taking place during meiosis. The answer is recombination. Crossing over means recombination takes place. Law of independent assortment is applicable for the traits which are located on different chromosomes, which are located on same chromosomes, which are located on homologs. The independent assortment, how the basic rule is, the traits should be located on different characters. If the traits are located on same chromosome, what will happen? They'll inherit together, so there won't be any crossing over. So that will be called as linkage. Next is the 3 important laws of heredity proposed by Mendel is related to segregation, independent assortment and law of dominance. Mendel did not propose laws of segregation, dominance, incomplete dominance and independent assortment. Answer is incomplete dominance. The reason behind the success of Mendel was choice of material, use of pure line, maintenance of qualitative and quantitative records. Test cross क्या होता है? It is used to investigate whether the dominant expression is homozygous or heterozygous. It involves mating of F1 hybrid with homozygous recessive parent. Okay. In F2 hybrid, to check the tall plant is homozygous or heterozygous, which cross is performed? That is nothing but test cross. बैक क्रॉस कर सकते हैं, लेकिन टू चेक एग्जैक्टली वेदर इट इज़ होमोजाइगस और हेटरोजाइगस, मेंडल यूज्ड टेस्ट क्रॉस एंड दिस इज वेरी प्रोमिनेंट वन. रेशियो रेफर्स टू बैक क्रॉस. बैक क्रॉस क्या होता है? F1 कैन बी क्रॉस्ड टू एनी ऑफ द पेरेंट. इट मे बी बैक क्रॉस क्या होगा? F1 विल बी हेटरोजाइगस. ठीक है, सो वी मे क्रॉस विथ एनी ऑफ द पेरेंट. That is nothing but your back cross. तो टेस्ट क्रॉस क्या होगा? F1 should be only be crossed to its homozygous recessive parent. A cross between individuals with unknown genotype for a particular trait with a recessive plant for the trait is called as test cross. Interaction between two alleles which are present on the same gene locus of two homologous chromosomes is called इंट्राजेनिक इंटरेक्शन एंड इंटरअलीलिक इंटरेक्शन. रिमेंबर, ये बहुत ही इंपॉर्टेंट क्वेश्चन है. इंटरेक्शन बिटवीन टू अलील्स, दो अलील्स है विच आर प्रेजेंट ऑन द सेम जीन लोकस ऑफ टू होमोलॉगस क्रोमोसोम्स. तो अगर दो होमोलॉगस क्रोमोसोम्स के बीच में टू अलील्स लोकेटेड है तो उसके बीच में जो इंटरेक्शन या क्रॉसिंग होगा वो इंट्राजेनिक इंटरेक्शन और इंटरअलीलिक इंटरेक्शन होगा. नेक्स्ट क्वेश्चन. Interaction between the alleles of different genes on the same or different chromosome is known as intergenic or non-allelic. RR, capital RR, red flowered plant of Mirabilis is crossed with small rr, that is white flowered plant of Mirabilis. All the capital R, small r offsprings are pink. This is an indication that R gene is incompletely dominant. ठीक है. Incomplete dominance, one could get 1:2:1 ratio in F2 generation. Codominant differs from incomplete dominance as in codominance both the genes express equally. For example, आपके blood group हो सकते हैं या रैबिट का कलर हो सकता है. इसमें सब क्या होता है? कोडोमिनेंस होता है, तो कैरेक्टर्स इक्वली एक्सप्रेस होते हैं. नेक्स्ट क्वेश्चन इज मल्टीपल alleles of a gene always occur on the same locus of a chromosome, the same position on different chromosome, different loci on a chromosome or different loci on different chromosome. आंसर क्या होगा? सेम लोकस ऑन ए क्रोमोसोम. मतलब मल्टीपल अलील्स एक ही लोसाई पे अपना पोजीशन एक्वायर करते हैं. इन ड्रोसोफिला द जीनोटाइप ऑफ नॉर्मल विंग्स VG सुपर ये M, then VG plus VG NO, then VG. नॉर्मल विंग का जीनोटाइप क्या होगा? VG प्लस. ठीक है. ABO ब्लड ग्रुपिंग इज बेस्ड ऑन मल्टीपल अलीलिज्म और कोडोमिनेंस. कोडोमिनेंस ज़्यादातर हम नहीं बोल सकते हैं. दिस इज ऑलवेज मल्टीपल अलीलिज्म. ब्लड ग्रुपिंग इन ह्यूमन इज कंट्रोल्ड बाय 3 alleles in which AB is codominant. In pleiotropic inheritance, different traits are controlled by single gene. Example of pleiotropy, sickle cell anemia. Genotype of a carrier carrying a gene for sickle cell anemia is HBA by HBS. ये हमेशा आप याद रखिए. Genotype of a carrier carrying a gene for sickle cell anemia is HBA by HBS. In which disease does the RBC of a person becomes half moon shaped? आंसर है सिकल सेल अनीमिया. इसमें हाफ मून शेप्ड ब्लड ये होते हैं क्या? आरबीसीस होते हैं, जिससे प्रॉपर ब्लड सर्कुलेशन में भी प्रॉब्लम होता है. मैरिज बिटवीन 2 कैरियर्स ऑफ सिकल सेल अनीमिया विल रिजल्ट इन टू 1 नॉर्मल एंड 2 कैरियर, 1 सिकल सेल एनीमिक, तो दोनों हो सकता है। When single character is controlled by 2 or more genes, it is called polygenic inheritance. When many characters controlled by a single gene, वो क्या होगा? प्लियोट्रोपी। मतलब प्लियोट्रोपी और पॉलीजेनिक इनहेरिटेंस दोनों एक दूसरे को ऑपोजिट हैं, ऐसे याद रखिए। नेक्स्ट क्वेश्चन इज द एडिटिव और क्यूमुलेटिव इफेक्ट इज शोन बाय पॉलीजीन्स। Who discovered polygenic inheritance? Nelson Allele. Phenotypic ratio of red and white kernel in F2 generation showing polygenic inheritance is, polygenic inheritance का ratio हमेशा होता है 1:4:6:4:1. When red wheat kernel is crossed with white wheat kernel, the probability of getting a red darkest plant is 1:16. तो एक ऐसे होता है इसका 1 डार्क रेड, 4 मीडियम डार्क रेड, फिर 6 मीडियम रेड, फिर 4 रेड एंड 1 वाइट होगा। ऐसे इसका रेशियो बांटा जाता है। द फेनोटाइपिक रेशियो ऑफ पॉलीजेनिक रिप्रेजेंटिंग स्किन कलर इन ह्यूमन इज 1:6:15:20:15:6:1। ये कुछ नहीं है, इसी का एक्सटेंडेड फॉर्म है, ठीक है। अगर आप पिरामिडिंग शेप में जाएंगे 1:1:2:1, फिर 1:3:3:1, उसके बाद में 1:4:6:4:1, फिर इसका नेक्स्ट वर्जन क्या होगा 1:5:10:12, फिर 10:5:1। तो ये रेशियो जो 6 है तो ये यहाँ पे आएगा, ओके। सॉरी, इसमें और पॉलीजेनिक रेशियो ऑफ स्किन कलर ऑफ ह्यूमन में कोई संबंध नहीं है। जो मैंने अभी पिरामिडिन शेप का जो मल्टीपल फैक्टर का एक्सप्लेनेशन किया था उसको और इसको रिलेशन नहीं है। दिस इज प्योरली द स्किन कलर फैक्टर which is controlled by polygenes, okay, that is 1:6:15:20:15:6:1 जो इसका एक्सटेंडेड फॉर्म है, ठीक है। फिर से आप यहाँ आगे बढ़ेंगे तो 1:6:15:20:20 ऐसे होते जाएगा, ठीक है। Who studied the inheritance of skin color of Negro and white population in USA? इसको स्टडी किया था डेवनपोर्ट ने। Skin color is controlled by 3 pair of genes. When a Negro married white, how many phenotypes are obtained? आंसर है 7, ठीक है। वो डिफरेंट कलर कॉम्बिनेशंस बनेंगे। कैपिटल A स्मॉल a, कैपिटल B स्मॉल b, कैपिटल C स्मॉल c is the phenotypic ratio of mulatto. The science dealing with the heredity and variation is known as genetics. The peculiar character of pea flower is papilionaceous corolla, जो सारे डाइकॉट्स में हम पा सकते हैं। To avoid the birth of child with fetal sickle cell anemia, क्या कर सकते हैं? Marriage between 2 homozygous is discouraged. Marriage between 2 heterozygous is also discouraged. नेक्स्ट, कैरेक्टर ऑफ माइटोकॉन्ड्रियल डिसीज़। माइटोकॉन्ड्रियल डिसीज़ जैसे आप जानते हैं ज़्यादा कैरेक्टर्स तो इनहेरिटेंस या हेरिडिटी फैक्टर पे डिपेंड होते हैं, जैसे कि क्रोमोसोम या जीन में प्रॉब्लम होते हैं तभी डिसीजेस आते हैं, या कोई भी पॉइंट म्यूटेशन होता है, फ्रेम शिफ्ट म्यूटेशन होता है, या टर्मिनेशन हो जाता है, इससे सब डिसीज़ कॉज होता है। तो थ्रेशोल्ड इफेक्ट, एक्स लिंक्ड कैरेक्टर्स, अफेक्टेड हार्ट ब्रेन मसल, ये सब माइटोकॉन्ड्रियल डिसीजेस के कैरेक्टर्स हैं। विच ऑफ द फॉलोइंग आर नॉट मल्टीफैक्टोरियल डिसऑर्डर्स? मल्टीफैक्टोरियल डिसऑर्डर्स कौन से हैं? जैसे कि सीजोप्रीनिया हो गया, क्लेफ्ट हो गया, स्पिना फेबिडा हो गया। लेकिन मल्टीफैक्टोरियल डिसऑर्डर जो नहीं है वो है क्राइडोकैट सिंड्रोम, क्लीनफेल्टर सिंड्रोम, जो सेक्स क्रोमोसोम होते हैं XX Y या कोई भी X डिलीट हो जाता है उस कंडीशन से ये सब बनते हैं जो मल्टीफैक्टोरियल डिसऑर्डर नहीं होते हैं। नेक्स्ट क्वेश्चन, गिव द डेफिनेशन फॉर uniparental disomy. When a person inherits two copies of a part of one chromosome from one parent and no copies from the other, example PWS. Which of the following is not often seen in the individual with Klinefelter syndrome? आंसर है webbed neck. The recurrence risk for trisomy 13 is increased by options: advanced paternal age, 13/15 translocation in one of the parent, extensive methylation of chromosome 13, advanced maternal age. आंसर होगा दो होंगे यहाँ पे, 13/15 ट्रांसलोकेशन इन वन ऑफ द पेरेंट, फिर एडवांस्ड मैटरनल एज. नेक्स्ट क्वेश्चन है विच ऑफ द फॉलोइंग विल यील्ड द हाइएस्ट रिकरेंस रिस्क फॉर डाउन्स सिंड्रोम इन ए फैमिली. द ऑप्शंस है 35-year-old mother, 65-year-old father, 25-year-old mother with a previous child with trisomy 21, then 20-year-old mother who carries 21/14 Robertsonian translocation and has had no previous children with Down syndrome. आंसर यहाँ पे होगा D. The clinical diagnosis of the child with a karyotype 46XY deletion is Prader-Willi syndrome. Next is the recurrence risk of trisomy 13 is due to Robertsonian translocation is 33% if one of the parent has karyotype 45. ये सब ह्यूमन जेनेटिक्स से रिलेटेड है, CSIR में पूछ सकते हैं, इसलिए मैं जस्ट डिस्कस कर रही हूँ, इतने इम्पॉर्टेंट नहीं है. The recurrence risk of thalassemia for the sibling of the affected fetus is 25%. Next is people with sickle cell trait are heterozygous for the sickle cell allele, are usually healthy, have increased resistance to malaria, produce normal and abnormal hemoglobin. A neural tube defect could be detected prenatally by ultrasound. टाइप 2 डायबिटीज़ कौन से होते हैं या कौन से नहीं होते हैं, यहाँ पे पूछा गया है. ऑप्शंस है टाइप 2 डायबिटीज़. दिस फॉर्म ऑफ डायबिटीज़ इज़ मोर हाईली हेरिटेबल देन टाइप 1. ससेप्टेबल टू टाइप 2 डायबिटीज़ इज़ partially due to the MHC and insulin genes. Diet and exercise can decrease susceptibility to this form of diabetes. MODY, that is maturity onset of diabetes in the young, is considered a highly heritable subset of this form. तो ये सब है टाइप 2 डायबिटीज़ के बारे में. तो यहाँ क्वेश्चन पूछा गया है क्या टाइप 2 डायबिटीज़ के बारे में नहीं है. तो टाइप 2 डायबिटीज़ क्या है? इट इज़ ए नॉन इंसुलिन डिपेंडेंट डायबिटीज़. टाइप 1 डायबिटीज़ क्या होता है? इंसुलिन डिपेंडेंट डायबिटीज़ होता है. तो आंसर है दिस टाइप ऑफ डायबिटीज़ टिपिकली हैज़ अर्लियर एज ऑफ ऑनसेट देन टाइप 1 डायबिटीज़. ये फॉल्स आंसर है क्योंकि जब इंसुलिन डिपेंडेंट होगा तो वो टाइप 1 काइंड ऑफ डायबिटीज़ होगा, where this type of diabetes typically has earlier age of onset than type 1. The crossing of F1 to any of the parents is back cross. ये सब हमने ऑलरेडी डिस्कस किया है. एक और क्वेश्चन है सिस्टिक फाइब्रोसिस इज ऑटोसोमल रिसेसिव डिसऑर्डर. 9:7 रेशियो इन द F2 जनरेशन रिप्रेजेंट्स इनकम्प्लीट डोमिनेंस, कोडोमिनेंस, एपिस्टेसिस, कम्प्लीमेंट्री इंट्रैक्शन. इसका आंसर होगा कम्प्लीमेंट्री इंट्रैक्शन. देन स्मॉल अमाउंट ऑफ लीथल म्यूटेशन इज ऑलवेज प्रेजेंट इन द पॉपुलेशन ड्यू टू म्यूटेशन सेलेक्शन बैलेंस. इफ ए प्लांट विद a genotype capital A small a capital B small b is selfed, the probability of getting capital A capital A capital B genotype will be 1/4. Sorry, 1/16. It is not 1/4. How many phenotypes can occur in the human blood group ABO with allele IA, IB, I? That is 1. विच टर्म रिप्रेजेंट्स पेयर ऑफ कॉन्ट्रास्टिंग कैरेक्टर? इसको अलिलोमॉर्फ बोलते हैं. True breeding tall plant was crossed with true breeding short. The result was all tall. Phenotype is expressed as the physical makeup. Evolutionary changes are always inheritable. That's all, friends. If you have any doubt, put it in the comment box. Thank you for your patience listening and watching.